Molecular biology of the prions. Of course, we have already studied that prions are the proteins. So molecular biology, biology of the molecules which are inert molecules, which are the necessary for the necessary for the animals, or necessary for the human beings, or necessary for the life, is called the molecular biology. So we will see that molecular biology from the molecular biology point of view, from the molecules point of view, what is the prions. Of course, they are the proteins, so we will see their structure, or we will see the from the molecular point of view how we can distinguish what is the proteins and how these proteins, when they are in the normal condition, how they will be able to abnormal, or how they will be able to cause some kind of the disease. So prion proteins, this is the abnormal isoform of the normal proteins. Located in the cell surface, of course, these are located which kind of the cells? Of course, the brain or the nervous tissue of the cells, these are located there. So whenever the disease is caused by them, the disease is caused by the disease will cause the harm to the nervous system, disease will cause the harm to the brain. Normally, the protein protects cell against the oxidative stress. Free radicals lacking from the electron, and that's why this is the normal function of the protein. So prion proteins, these are the, we will conclude from this that they are the normal isoform of the normal protein, abnormal isoform of the normal proteins, and they are located in the cell surface of the brain and the brain tissues, and these are the protecting the cells against the oxidative stress. There are two kinds of the proteins, one is a normal protein and other one is the abnormal protein. PRPC is the cellular protein, this is called the cellular protein, so we will denote it as PRP in C. This is the protein, prion protein, which is in the cellular form and which is also the normal proteins. So this normal protein will be converted into the abnormal form in the kind of the PRP SC. SC is denoted for the Scrappy form. So it will be from the C cellular form to the Scrappy form, and this is the infectious form, and this is the abnormal form of the isoform of that normal protein. And if the presence of the PRPC will cause the disease, and presence of the PRPSC will cause the disease, and PRPC is normally present and it will not cause the disease, it will normally present on the body, on the cell surface. The molecular biology of the prions. What are the prions from the molecular biology point of view? Prions, the PRPC is the cellular form, is the normal form, and the PRP scrappy form. These are the two forms. We have given them in the tabulated form here, and we can differentiate both of them from each other, that how they are different from each other. First of all, the PRP cellular, and these are rich in the alpha helix. So whenever we see the shape, whenever we see the structure, and we will see from the molecular point of view, we will see that the PRPC is the helices. There the alpha helices are present in there. But whenever it is converted into abnormal form, it is very common to see the beta sheets there. There are about 43% of alpha helices here, and then there will be the beta sheets more as compared to the alpha helices in the structure of the PRPSC in the Scrappy form. The cellular form is of course the soluble. However, the abnormal form, the Scrappy form, will not be soluble and will be the organic. Then the protease PK protease sensitive PRPC, while the PRPC, the Scrappy form, will be the resistant. Definite shelf life, the PK sensitive will have the definite shelf life and then degrades when no longer needed. In the normal isoform, the PK protease sensitive will always present. They will have the shelf life, they are the sensitive, and they will be present in the definite shelf life whenever their life exists, and they will degrade when they are not needed in the body. However, in the PRP scrappy form, they are PK resistance. No aggregation in the PRP normal form, cannot polymerize into the aggregates. However, on the other hand, when it is converted into the abnormal form, there will be the aggregation will occurs, which is easily occurs, and allows polymerization to form the fibrils, and similarly due to the beta sheets the aggregation will be occur. So these are the some specific ways, specific abnormalities which we can show from the molecular point of view of the prions, that they converted from the normal form to the abnormal form, and then this will be cause the disease. This is the normal form, and the aggregation and the non-aggregation because of this disease on the brain tissue will cause the sign and symptoms we see because of this disease, and that's why this disease is causing that because of this abnormality form. Here we can see the structures which we have studied, that it has the alpha helices. PRP cellular form will have the 43% of the alpha helices, while when it is converted to the abnormal form, the alpha helices will be reduced and they will be about 30%. We can see the alpha helices, and at the same time there are the present the beta sheets, which are the causing the abnormalities, and they are about 43% we can see in the abnormal form.